Sugar, what do we know about it? It's sweet, it's granular, and it's more addictive than some drugs, so got a lot going for it. But they're just general things. In scientific terms, we need to know that sugar is actually a polymer, much like we discussed earlier, and is made up of lots of little monomers called saccharides, monosaccharides to be precise. I'm sure you've heard of the term glucose, which is a term usually used to describe sugar, but it's not the only type. You also have things like fructose and galactose. These all fall under the term monosaccharide, which means a single sugar. Now, the term carbohydrate comes from the structure, the actual chemical structure of these molecules. When we look at them at this molecular level, you see that they're made up of carbon, hydrogen, and oxygen. When you hear the term 8 in a chemical, it just means that oxygen part, and then carbo is carbon, hydro is the hydrogen, and then 8 would be the oxygen. Now, I'm sure you're all looking forward to the reaction that takes place between two of these to form these long carbohydrate chains. And as stated in the previous video, bonus points if you've already got it, these are called condensation reactions. So what these condensation reactions do in terms of glucose is, at the ends you can see the hydroxy groups and the hydride group. So what it does is one of the hydroxy groups will come off and be removed along with one of the hydride groups, which forms a H2O, and then a bond will form between them, making what's called a disaccharide. In this case it would be maltose, two glucose molecules put together, but in the grand scheme of things, what would happen is this long chain would continue to grow and grow and grow. And due to the way that it's structured, it's obviously not a straight chain, and it would form a spiral known as amylose. Now, amylose is one of the sections of starch, which we'll come to learn about in a bit. Glucose itself is one of the easiest carbohydrates for us to use in our bodies, mainly because our system is able to dissolve those molecules and pass it through the bloodstream quite easily. Now, glucose comes in two isomers that we have to worry about, and they are alpha glucose and beta glucose. Whenever you hear the term iso, it simply means the same, and in the case of isomer, it's simply the same molecular formula with a different structure in some part of it. In the case of alpha and beta glucose, the difference is that the hydroxy groups are either on opposite sides of the structure or on the same side of the structure. Now, alpha has it so that the hydroxy groups are on the straight side, whereas beta glucose has it on both sides. But effectively what happens in biology is plants will use beta glucose and animals will use alpha glucose. Beta glucose is used a lot in cell walls for the plants. It helps give them a stronger structure between layers of those glucose molecules because they form straighter structures that pack nice and tightly on top of each other, creating this rigid structure. If you ever go outside and you see a plant, I know it's probably not the best time to, but if you do and you find a leaf, I'm sure you've all seen a leaf before, that structure itself will be made up of tiny molecules of beta glucose all joined in a long chain forming that cell wall. Let's now talk about disaccharides. Disaccharides aren't these long chains of polysaccharides, poly meaning many, saccharides meaning sugar. They're not long chains, they're rather two separate molecules that have been joined together through this condensation reaction to form individual double structures. Now, if we go back to the three structures that I told you about, glucose, fructose and galactose, we can begin to join two molecules together and find what we get out of it. So for glucose and galactose, we put that together and we get lactose. And I'm sure you've heard of lactose in things like milk. The other structures that we can make are glucose and fructose to make sucrose. You might have seen sucrose in sweetener packs that are designed to be put in drinks and things of that sort. So I'm sure you're wondering what we get if we cross galactose and fructose. The only thing is, these molecules will not naturally go together. You can also bind together two glucose molecules, as we said before, and if you remember from the previous section, we can make a structure called maltose. Maltose is used quite a lot in artificial sweeteners, but the main structure that it comes from is starchy foods or grains that have been processed down to get this maltose. So from here we know that condensation reactions form these longer chains, and when we have a lot of condensation reactions of alpha glucose, we form these starch chains, long spirals made out of amylose and amylopectin, which is another form of condensed glucose which has a more branched structure. When these two glucose molecules bind together, they form a bond that's similar between all saccharides, and it's called a glycosidic bond. Each molecule will have a certain bond that's formed between them, not necessarily different in chemical properties, but it certainly gives you a respect for what structure is going to form when these bonds are present. So if you're told in an exam that a structure has many glycosidic bonds, you're not going to think proteins, you're going to think sugars. Now, the final part. How do we know if the substance that we have in front of us contains any form of glucose? If we're looking for glucose independently, it's quite easy to do. All we do is we take the food sample, we process it to the point where we can dissolve it in water, then we place it in the water along with a solution called Benedict's solution. Benedict's is also kind of known as Fehling's in chemistry, but we don't need to know that for this particular subject. When we've added this blue solution in, we place the boiling tube into a water bath at quite high temperatures, which overcomes the activation energy for this reaction to occur. And what we'll see is, over time, the chemicals inside of the Benedict's solution will be reduced by the glucose molecules, and because of that we call glucose a reducing sugar. As far as the other two chemicals go, fructose and galactose, we have the same chemical reaction occur. However, the reducing effect on this Benedict's solution isn't nearly as much as the glucose that affects it. The overall reaction for glucose sees the solution turn from a blue to a brick red. That's an important term to remember, is that brick red helps discern from other types of sugar present. But the other two solutions will push the Benedict's towards a orangey colour, maybe a yellowy colour, and it won't nearly be as deep red as this brick red that the glucose has formed. Finally, the disaccharides that we mentioned before don't have a reducing effect on the Benedict's, so instead we have to break it up into the two constituent parts. The way we do this is we take the sample of food that we've crushed up and we place it in the boiling tube again. We pass some hydrochloric acid into the mixture and wait for that to break down. We wait maybe 5 minutes to make sure everything's completed. We then take this hydrochloric acid solution out and we pour in some basic solution. Some sodium hydroxide usually works, and that neutralises the acid so that we know when we put the Benedict's in that it's not going to be the acid that's reacting with it, it's going to be the sugar and only the sugar. We make sure that we put the same amount of moles in of base as we do with acid, and that makes sure that there's no excess of anything. So what happens when we add our Benedict solution in is the free-floating molecules of monosaccharides will reduce that Benedict's. It's just like a spectrum of light. The deeper red it goes, the more reducing that sugar molecule is.